Good morning to all. Today I'm talking about the sterilization and disinfection. You all of them know about the sterilization. And first I have to talk about the definition of sterilization. Sterilization is a process by which an article, surface, or medium is free of microorganism, either in the vegetative form or in the spore form. And then disinfection. Disinfection is destruction of pathogenic microorganisms by using chemical agents. Some other words are asepsis, and aseptic techniques are mainly used to prevent the contamination of the surgical instruments and then patients during surgery. Then bacteriostatic are there, bactericidal are there. Static means the inhibiting agents are used for the bacteria, that is called as bacteriostatic agents. That the agents are used to kill the bacteria is called as bactericidal agents. The classification of sterilization are mainly physical methods and chemical methods. Physical methods are mainly sunlight, drying, heat. Heat are mainly dry heat is there, moist heat is there, radiation or non-ionizing radiation and ionizing radiation, and filtration. Chemical methods of sterilization are called as disinfection. They are alcohols, aldehydes, dyes, halogens, phenols, surface active agents, gases, and metallic salts. The main physical methods are coming to the details. Their physical methods are mainly sunlight. Sunlight, you already know about the sunlight. They are the natural spontaneous activity. Then germicidal and microbicidal activity. UV rays are there, and then UV rays are because of the UV rays, which the sunlight have the germicidal activity. Cidal means the killing, and microbicidal means killing of the microorganisms. Mainly, the natural methods are mainly used for the drying of clothes. And drying, the moist reduction, the 3/4 of the bacteria contains the water. The moist reduction produces the deleterious effect of the bacteria and the killing of the bacteria. Then heat are mainly dry heat and moist heat. I already showed in the previous slides. Then dry heat, they are the principle behind the dry heat is denaturation of the proteins or coagulation of the proteins are happening, and then because of the denaturation, the bacteria is killed and oxidizing the chemical constituents. They are the two principles behind the dry heat method. Dry heats are applying in 4 ways. They are red heat method, and then incineration, hot air oven, and flaming. First one is red heat method. The main uses of red heat method are for the sterilization of the inoculation loop or wire, tip of the forceps, and spatulas. And then how we have to do the dry heat means red heat method are all the instruments are held in the Bunsen flame till they are became red hot. The another method is incineration. Incineration is direct burning of the contaminated materials, that is 800 to 1000 degrees Celsius at the high temperature. And the main uses are the contaminated clothes, animal carcasses, and pathological materials. Flaming, the red hot and flaming, only some differences are there. The passing through the Bunsen flame, just will pass through the Bunsen flame without allowing them to become a red hot. That is called as flaming. The main uses are for the sterilization of the scalpels, needles, and glass slides. Then next one is hot air oven. This is the widely used common methods of sterilization. And then structure of the hot air oven are the 2-wall chamber. Inner wall is composed of copper and outer wall contains the asbestos. And chamber are divided into the small compartments with the help of removable racks. Thermometer are inserted at one of the corners, and it's heated by electricity. Heating elements in the wall of the chamber, and fan is fitted in the distribution of the air. And then fan is the distribution of the air through the elimination of through the air pockets. And structure of the hot air oven. Then main uses of hot air oven are to sterilize the forceps, scissors, scalpels, and swabs. And some pharmaceutical products are also sterilized by this method: liquid paraffin, dusting powder, fans, and things. Next, sterilization control. You all of them know about how to check the efficiency of hot air oven. The normal temperature of hot air oven is 160. The halting period of hot air oven is 160 degrees Celsius for 1 hour. And the biological method are used to check the efficiency of hot air oven is not toxigenic strains of Clostridium tetani. Clostridium tetani have the spores. Then we have to take the spores and impregnate it with the pepper strip and keep in the hot air oven. The halting period is 160 degrees Celsius for 1 hour. We have to keep that temperature, and then after the sterilization, take the pepper strip, take the spores and inoculate it into a suitable medium. Clostridium tetani suitable medium is an RCM, Robertson cooked meat medium, inoculated, incubated. Any growth is there, and then it's not an efficient method. The another one, brownish tube. Brownish tube have one small hot small red color is there, and then that red color became green color. Green color means it's not an efficient method. And thermocouples are the temperature are recorded by these thermocouples. Next one, moisture sterilization. Principle behind the moisture sterilization are the microorganisms are killed by coagulating the proteins. Then different moisture sterilization methods are the temperature below 100 degrees Celsius, temperature at 100 degrees Celsius, steam at atmospheric pressure, steam under pressure. Temperature below 100 degrees Celsius, what of the methods is? One is pasteurization, INSPISSATOR, water bath, and vaccine bath. The pasteurization are mainly process of killing of pathogens in the milk. All of them know about pasteurization. Two methods are the Halter method and then flash method. Halter method is milk is heated at 63 degrees Celsius for 30 minutes, and flash method is 72 degrees Celsius for 15 to 20 seconds. Next one is water bath. Water bath are actually by water the temperature is special water bath temperature is 56 degrees Celsius for 1 hour, and then vaccine bath are 60 degrees Celsius for 1 hour. Next I am talking about the INSPISSATOR. The INSPISSATOR, the temperature for INSPISSATION is 80 to 85 degrees Celsius. It's mainly used for the sterilization of some special media, that is Lowenstein Jensen medium and Loeffler syrups. No? That mediums are sterilized by this the less temperature, that is in 80 to 85 degrees Celsius for half an hour for 3 successive days. What is the principle behind the INSPISSATOR means? The first day of sterilization, mainly we are using for the sterilization of the media, but the first day of sterilization, we are sterilizing the vegetative forms of the organism, whatever the vegetative forms of the organism are present in the medium. The next day, any spores are there that will be germinated, and it is in the form of a vegetative cells, and that is killed by the next day. And any remaining spores are there that also will be germinated, and the third day of sterilization will sterilize all the medium. That is the principle behind this INSPISSATOR. Then next one, temperature at 100 degrees Celsius. Mainly the temperature at 100 degrees Celsius are boiling. Boiling, the important methods of coming under this boiling is tyndallization or intermittent sterilization. Tyndallization is done by using the steam sterilizer, and that is steam at atmospheric pressure. It's by Koch or Arnold steamer, consists of a tin cabinet, and it is a conical lid to enable the drainage of the condensed steam. Perforated tray above ensures materials are surrounded by the steam, and for the routine sterilization, that is exposure of 90 minutes is used. The process are done by using steamer, that is called as tyndallization or intermittent sterilization or fractional sterilization. The same procedure of the INSPISSATOR, that is also for 100 degrees Celsius for 20 minutes for 3 successive days is used. And then intermittently we are doing, because we are using the 3 days or fractional days we are using for this. That's why it's called as an intermittent sterilization. Thank you.